Hello, my name is Igor Grigoriev. Thank you for joining me today. It's a little bit unusual for me to give a talk to a virtual world when I don't see the audience, but I hope you're all listening, and if you listen and if you have a question during the course of my talk, you can click on the button Q&A in the lower corner of your screen and type me a question, and by the end of the talk I hope to address most of the questions that I will receive. Today I would like to talk about future. It will not be connected to human health, but it will be important in relation to human health. It will be about health of planet. If you look at my first slide, you see two options we may have for the future. One on the right side is bright, sunny, and clean water, and one on the left side is a little bit darker. And unfortunately, we have choice of either of them in different parts of the world, and these are two options for our immediate future, and they're directly related to energy and environment challenges that we're facing right now. I believe strongly that biology, and in particular genomics, offers tools that would help us to better understand the processes going on in nature and help us to shift the balance towards a brighter and hopefully happier future, which we see as an example on the right side. So I work for the Joint Genome Institute, where I lead Fungal Genomics program. And hopefully by the end of this talk I will convince you that fungi play an important role in nature, and understanding genes encoded in their genome will help us to control our future. So Joint Genome Institute, let me start with a couple of words about Joint Genome Institute, or JGI. It's a genomic center. We're located in Walnut Creek. Here you see a map. A red dot on the map indicates our location. We're located in Walnut Creek, not far away from San Francisco. It is in California. But we are also Department of Energy Genomics User Facility. Probably the only genomics facility in the world devoted to the problems of energy and environment, while there are many centers devoted to human health. And this map also indicates users, different groups of researchers from all over the world, not only from the United States, that currently work with us on different genomics projects and trying to answer different types of questions. Joint Genome Institute started back in 1999 with the idea to contribute to the Human Genome Project, which was very active at that time, and we successfully finished and annotated 3 human chromosomes. And since then, our main focus was on missions of Department of Energy, which are bioenergy, carbon cycling, and biogeochemistry, the 3 major areas where different questions could be addressed with genomic science. As I mentioned, we started with Human Genome Project, and that was high-throughput sequencing on Sanger machines. And we live in an era of genomics revolution, where every other year we have a new sequencing platform, which leads to avalanche of genomic data that need to be interpreted. And we tried different platforms. There are 4x4, Illumina, Pacific Biosciences, and others that you name. And combination of the couple of those right now give us ability to produce a broad spectrum of products, starting with sequencing reference genomes, transcriptomes, analyzing expression of genes on different or fungi growing under different conditions, and not only fungal genomes, of course. Look at population questions, resequence, either natural variation or engineered strains. And all this huge amount of data produced by these sequencers are then analyzed with various analytical pipelines that run on supercomputer, or on the various computational clusters in Supercomputer Center in Berkeley, which NERSC is our partner. And another thing that we're doing, and which is our strategic goal, is to go from sequence to the function. With this explosion of sequencing techniques, we and other different centers produce huge amounts of data. We have tons of sequence, but very little is known still about function. And we'd like to bridge this gap between the large number of sequences and very small number of functions by developing high-throughput techniques for functional genomics. Starting from transcriptomics, ChIP-seq, proteomics, and others. In the world of fungal genomics, the program that we started about 3 years ago, at this point we contributing to more than half of the projects around the world, fungal genomics projects, according to the GOLD database, which is trying to keep track of various genomics projects happening in different parts of the world. All this sequence data is then processed, genomes are assembled, annotated, and integrated with various analytical tools on the portal, web-based portal called Microcosm, which is available at this web address you see on your screen, jgidu.gov/fungi, which currently has over 250 sequenced and annotated fungal genomes. And this number is growing very fast. To make sure that the data we produce and the tools that we also deliver are used, we focus our efforts on user support. And that varies from participating in a meeting like that and making sure that this genomic user facility is available for anybody around the world to sequence various genomes basically for free. And for sequence genomes that were proposed, we would like to make sure that the group of people who work on the genome effectively use the tools. We organize workshops in Jamboree to discuss findings coming out from analysis of sequences and annotated genomes. The value of fungi could be seen in various different ways. So here's an example of a dish that was produced by various fungi. First of all, it's mushrooms, as you see on the top. Then the bread produced with the help of bakery yeast. Then the cheese, where fungi play an important role as well. Even the little plant that you see on the top most probably cannot grow without symbiotic association with mycorrhizal fungi. And what you don't see there, a whole variety of drinks, from beer to wine. For fermenting, you require fungi as well. So fungi are important as a food. On the other side of this picture, you can see these relatively scary images of different diseases that caused by fungal pathogens of human, well, that create a basis for imagination for stories like X-Files to scare people a little bit further. But this is a reality, and the reality that drives development of different drugs. On the other hand, fungi produce drugs starting from penicillin, And, well, these basically several examples to illustrate how fungi are important in our day-to-day life, but these are not the reason why Department of Energy is interested in fungal genomics. For that, there is energy-related perspective. Interestingly enough, fungi are very important for growing biomass and converting biomass into biofuel. On this slide, you see how, well, at different stages of that process, different types of fungi are playing a critical role. For example, in order to get a sustainable growth of plants, you need to know everything about various plant diseases and explore fungi as pathogens, as well as understand symbiotic interactions between plants and, for example, mycorrhiza that deliver various nutrients for plants to grow. Once you're capable to produce sufficient biomass, it's need to be converted into sugars and then bioethanol. And fungi are the organisms that know how to overcome the barriers that plants develop during their evolution, lignocellulose that compose plant cell walls, and not only help plants to grow and protect them from microbial attack. Well, once you convert lignocellulose into simple sugar, again, fungi such as yeast can further ferment that to ethanol. And in addition to that, fungi are very important hosts in industry. They're used as producers of different enzymes. They are potential core machinery in biorefinery, which you can think about it as a factory converting biomass into biofuel, as alternative to oil refineries that are used currently. So in order to understand different aspects of how fungi operate in their different roles, we need to sequence their genomes. And fungal history of JGI started around the time when we finished with the human genome project. Back in 2004, the first fungus, the first JGI fungus was sequenced. It was the first Basidium acid, white-rot fungus for Nerpetic Rhizosporium. It was a big deal, and you see the image of this fungus on the cover of Nature Biotechnology Journal. And since then, there were many publications where we presented the first of its kind fungus, Silos fermenters, which I'll touch on in my talk, both pathogens and symbionts of poplar tree, another type of wood decaying fungus, brown rot, thermophilic fungi that are important for industry. And at some point, thinking about this individual story, we come up with the idea that instead of looking at individual genome, we should look at the group of organisms that show similar traits. For example, look at all symbionts or all pathogens as a group, sequence genomes of diverse representatives of this group, and compare them side by side and try to align the traits that are common between them with the genes that are shared. That gives a start to a project which called Genomic Encyclopedia of Fungi. This is encyclopedia again for energy and environment related questions. And instead of individual publication, we're thinking about chapter. Chapter is focused on plant health, chapter focused on biorefinery, and chapter focused on fungal diversity. I mentioned that JGI is a user facility, and every year we have a call for proposals. Anybody can apply to that. Basically through this call, we can open new chapters in this encyclopedia. But right now, for the rest of my talk, this will serve as an outline. I will talk a little bit about different fungi involved in plant health-related questions, then a little bit about biorefineries and fungal diversity. Let's start with the first question. Several years ago, we sequenced and published the genome of the first tree poplar, and also start looking at microbes and fungi associated with this tree, so that we build, well, sort of minimal ecosystems. So symbiont, Vaccaria bicolor associated, mycorrhizal fungus associated with the roots of poplar, and pathogen of poplar leaves. Besides interaction of fungi with its host, with its plant host, we also would like to explore questions of different lifestyles that we observe in the kingdom of fungi: symbiosis, pathogenesis, and saprotrophy, ability to live on a dead matter, and see what is common within each of these groups and what is different between all three of these groups. Starting with one of the examples, pathogen of poplar, Noemsera larissipusini. It's also called the poplar rust. It's a project we worked on together with a Broad Institute who explored another rust, wheat rust, and sequencing the genome sequencing of both genomes produced really large assembly. If the average size of fungal genome assembly is about 40 megabases, so these two fungi give us close to 100 megabases. They were full of transposons. About approximately half of the genomes were transposable elements. These are obligate parasites. They can live only inside the plant they infect. And the reason for that, based on analysis of genome, was broken metabolism, lack of certain genes that are required for the core metabolism. But the most interesting finding was a huge expansion in the number of genes coding small secreted proteins, which we think are critical in communication with the plant host, in controlling plant host, and all the very same proteins and the distribution of these proteins are shown on this diagram. So those that in red were the most expressed proteins when we looked at transcriptomics, expression of genes during leaf infection. So these small secreted proteins are very important for pathogens. In comparison, looking at the sort of opposite lifestyle, symbiosis, we can think about Mycorrhizal, the fungus that enter very close interactions with plant roots. So you see little plant, its root system, and these dense patches of association between plant roots and fungal mycelium. And again, a lot of signaling is involved. So the first Mycorrhizal fungus, Vaccaria bicolor, was sequenced. It was also relatively large, had a large number of different genes and over 500 small secreted protein. The most overexpressed gene in this genome was small secreted protein that was called MIP7. It's shown over 20,000-fold overexpression in ectomycorrhizal tissue in comparison to free living fungus. And further analysis of this little protein indicated that this gene is mostly expressed. You see this image on the right, which in green shows expression of this gene, and it's expressed in a space between the plant root cells, indicating that it's expressed right there during association of fungus with the plant, and this is a very intimate interaction, and the network of fungi is called Hartig network. Basically, it's very close interface between fungus and plant. So this is one side of symbiont. close signaling and close interaction with its host. On the other hand, it's still a decomposer. It's capable of decomposing dead material, dead organic matter, and for that we explored enzymes involved in this process called carbohydrate active enzymes, or enzymes for brevity. And we're surprised that in comparison to other fungi of different lifestyles, Laccaria bicolor have the smallest number of genes, almost no genes involved in decomposition of plant cell wall. So the first idea was that probably evolutionary, this organism eliminated all the genes that could potentially trigger plant response, because plants try to defend themselves from invasion from pathogens, and Laccaria bicolor may try to eliminate all of them. So in order to better understand this, let's look closer at different type of fungi, saprodes, and in particular wood decaying fungi. I mentioned that lignocellulose is a material that builds plant cell walls. It's composed of cellulose, which is this chain of glucose units, which are then wrapped and connected with each other by molecules of hemicellulose and wrapped in lignin. And all this is protecting cellulose from microbial attack. I also mentioned that fungi are among those organisms who know how to overcome this barrier. And the very first fungus that I already mentioned, Phanerochaetic chrysosporium, that causes white rot decay, which you see on this little image, its genome encodes genes that are critical for decomposition of both cellulose and lignin and hemicellulose. And we found a broad cocktail of enzymes involved in degradation of all these biopolymers. A few years later, we sequenced another wood decaying fungi, Osteoplacenta, which is a different type of rot. There are two major types of wood decay. One is called white rot, that causes degradation of all three components, lignin, cellulose, and hemicellulose. Brown rot is capable to degrade cellulose and hemicellulose, but not lignin. And that's what we've seen after sequencing the genome, no enzymes involved in lignin degradation. To our biggest surprise, we also didn't find the enzymes that were involved in degradation of cellulose. None of the genes we found in white rot were found in brown rot that are responsible for this process. But instead, in combination with transcriptomics and proteomics data, we found indirect evidence that maybe an alternative mechanism called phantom chemistry, which produces hyperactive hydroxyl radicals. So this is a non-enzymatic way to attack molecules of cellulose and decompose it, to degrade it to small units and ultimately to glucose. So that was 2 points of data sampling, 2 genomes of wood decaying fungi, and 2 completely different mechanisms encoded in each of these genomes that give us ground to start sequencing more. That's what we did, and last year we did a study. We published a study in collaboration with David Hibbett from Clark University and his colleagues, where we compared side by side not 2 but 12 different wood decaying fungi, and they're shown on this diagram in brown rods for brown rot, in brown rectangles for brown rot fungi and white rectangles for white rot fungi. So this much larger amount of data allowed us to look at different genes and different processes a little bit closer, and what's more importantly, in evolutionary context. Having all this whole genome data, we could reconstruct phylogenetic relationships and reconstruct evolutionary paths and reconfirm the idea that was expressed earlier, that probably both types of wood decay emerged from white rod's ancestor. Again, this data, using techniques called molecular clock analysis, we were not only able to reconstruct the ancestor and also look at approximately at the time when it's emerged, when it's evolved, and estimate that that's happened probably around 300 million years ago. Interestingly enough, around the very same time, according to geological records, the amount of organic material converted to coal suddenly reduced. So if you can think about history in terms of millions of years of you probably remember that between 400 million years ago and 300 million years ago, the planet Earth was covered with prehistoric forests. Trees were living, growing, dying, and ultimately converted into coal. So lignin is a precursor of the coal, and probably the very first white rod fungus figured out ways how to enzymatically process lignin, and basically could be potentially a reason that will lead us in some future to a coal crisis. But at the same time, the very same fungi keep secrets that could be used by us in order to understand how to process biomass and produce fuel for our needs using alternative approaches. So that was a little story about food decay, and let's go back to symbionts. Interestingly enough, you always see on all these trees, I'll go to the previous slide, you always see on these trees Laccaria as a member of this Agaricomycete, this group of fungi, and we looked at enzymatic profile. The number of enzymes is smaller than others, but we also see a reduction similar to brown rods. There are evidence that brown rod fungi, which can destroy our homes, can also grow and wrap the root system of different plants in a way similar to Mycorrhizal, symbiotic fungi. And under the microscope, we can also see that fungal cells get into very intimate interactions with the plant root cells, but they not form in the network as we've seen for symbionts. Interestingly enough, on phylogenetic tree, very often we see brown rods next to Mycorrhizal fungi. Those are marked in brown and oranges, and very often they're sister groups, meaning that they have the same. They evolved from same ancestor, and probably some switch, some loss again of some group of genes led to these different types of life that they live. And further analysis, more recent data indicate metabolomics analysis of small components and molecules they produce, also suggests that maybe some of the Mycorrhizal genomes encode genes that may be conducting the very same phantom chemistry that brown rods are doing. So that's very interesting and suggests sampling symbiotic fungi at much larger scale. And this is exactly what we're doing. One of the projects initiated by our collaborator Francis Martin from INRA from France, he, with a large group of researchers around the world, is looking at sequencing 30 mycorrhizal genomes. And on this picture, you see illustration on a petri dish scale. You see illustration of what's happening in nature. Interestingly, about 80% of plants form mycorrhizal association, and many of them cannot live without such association. The reason for that is mycorrhiza is capable to transfer nutrients to the plants, such as phosphorus, and without which plants cannot grow. What's interesting on this picture is the amount of green material. It's still in the early stages, but the amount of green material is much smaller than what you see under equivalent of what you see under the surface, within the soil. And that reflects the situation in nature. It's known that forests sink for carbon, if you think about carbon cycles, but recent study indicate that a lot of that carbon goes into roots and the fungal material associated with the roots. So at this point, we have about 20 genomes sequenced. They represent different types of mycorrhizal association. We were talking about Vaccaria bicolor, or ectomycorrhizal association, which you see in the middle, where fungal material grows in between plant root cells, which are these blue rectangles. And there is another, maybe more broadly, another type of fungal mycorrhizal association called arbuscular, where fungi grow into plant cells. And there are other types called ericoid and orchids, where a combination of both can be found. On phylogenetic side, there are organisms that are known to be mycorrhizal that happen in different groups of fungi, suggesting that this lifestyle evolved many times in the history of fungi. And so that's hopefully, right now we have about 20 genomes. Their comparative analysis indicate that there's a diversity of gene sets that may correlate with different lifestyles. I mentioned small secreted proteins for Vaccaria. The second sequenced mycorrhizal fungus, or truffle actually, done by the same lab of Francis Martin, indicated there are no small secreted proteins. It's organs belonging to a completely different group. And this whole spectrum of genes encoded in 20 genomes kind of fall into these groups, those that interact with plants by small secreted proteins and other types. To summarize this part of my talk, I would like to again highlight that, well, if we look at what we're trying to do is to look at individual groups of pathogens, symbionts, saprobes, find features shared between them. And to our surprise, there is more diversity within the group than similarity between the groups. We talked about very different ways of degrading wood encoded in genomes of brown and white rot. We talked about similarities between mycorrhizal symbionts and pathogens. And there are organisms that have white rot on one hand and pathogen of different trees on another. So it's hard to draw clear lines between them, but hopefully I illustrated how genomics can give you some ideas of what's going on in these different types of fungi. But the next step in this process is functional genomics, to understand what each of these genes do and how they interact with each other and how they're regulated within each of these genomes. With that, we go into the next more applied chapter of this encyclopedia called biorefinery. But the goal of this project is a little bit more pragmatic, a little bit more applied. We would like to come up with a collection of genes that could be used by industry, with a collection of pathways encoded in the sequenced genomes, and with a collection of hosts, or at least explore the hosts that are currently used for different purposes. And think about it as a list of parts, bioparts from which biorefinery or other application and machinery can be constructed. I mentioned that's why we're doing this large-scale sequencing of wood decaying fungi, give us the largest and most comprehensive collecting of food decay and enzymes. And the only thing we have at this point is predicted function for many of them. In order to understand what they do, we need to develop a model. Currently, there is a couple of well-established models in the fungal world: Saccharomyces cerevisiae yeast and Neurospora crassa filamentous fungus, but they all belong to the group of ascomycetes, which do not have wood decaying fungi, which have different type of symbiotic interactions with plants. And in order to understand basidiomycetes and processes encoded there, we would like to pick one of the organisms here. And one of those models is a genome that we also sequenced at JGI, the genome of Schizophilum commune, which is a model mushroom, which is an organism that was used to understand how mushroom is formed. The way to do it is to take a wild type strain, do some mutations, knock out transcription factors, for example, and see how you can grow this mutagenized fungus only to a certain stage, that, for example, it cannot grow fully developed mushrooms. And by combination of this experiment of different knockouts, you can identify all key transcription factors that regulate the processes of mushroom development. You can take those mutants and run transcriptomics analysis and see how that's affect genes downstream that's regulated by those transcription factors. And from that model, the cascade of interactions between different transcription factors and other genes. So going through the process of sequencing, identifying function of individual genes or groups of the genes, and then modeling the processes encoded by those genes. Right now, this probably is the only one of the few basidiomycetes that can be transformed and where those mutations can be done. And we're currently growing this at JGI to understand how this fungus grows on different, this is a white rot fungus, how it grows on different types of different carbon sources, on glucose, cellulose, different types of food. And using similar approaches, try to understand which transcription factors are critical to switch from one type of food to another for this fungus. And ultimately, well, delete genes, overexpress genes from other organisms and see what combination of, what cocktails of enzymes are more efficient for different types of food and how they're regulated. So that was an example of host or model we're using. And talking about pathways beyond individual genes, I would like to mention experiment, a collaboration with Great Lake Bioenergy Center, where we looked at the next step after decomposition of lignocellulose, converting sugar into ethanol. Saccharomyces cerevisiae, baker yeast, can easily convert glucose into ethanol. And at the same time, probably about a quarter of biomass, plant biomass is composed of xylose, different types of sugar, which is uniquely processed by fungi inhabiting guts of beetles that live on different plants, wood-eating beetles. The fungi that grow in absence of oxygen, and sequencing of that type of fungi, first of which was Pichia stipetis, we identified and were able to reconstruct the pathway that leads not only from glucose to ethanol, but from xylose to ethanol, identified genes that are critical for that, that allowed other people to try to integrate those genes into into genome of Saccharomyces cerevisiae to make that organism convert not only glucose but xylose, convert all sugars all measured sugars that we can obtain from plant material. So next, that happened, and performance of that modified Saccharomyces cerevisiae wasn't the best. That triggered the next step of development, and looking closely at the groups of organisms that can consume xylose and convert it into ethanols. They are marked here in blue. We have 3 of them, and compare them and contrast with those that are purple, which can consume xylose but do not produce ethanol, and with all other outgroups. So we have many tools for these types of comparison. So we can look at the genomes and see what's common between blue genomes and purple genomes. We can look at transcriptomes and see what is the most expressed genes in one group versus another, and combine all these potential candidates for further improvement of Saccharomyces cerevisiae production strain. We come up with a subset of genes, and our collaborators at Great Lakes Bioenergy Center are working on integrating them into Saccharomyces cerevisiae and showing that improved versions have improved xylose utilization, better growth, and ultimately better production of ethanol. So this is an example where you can go from genome sequence and through comparative analysis, transcriptomics, and other experimental techniques, identify genes that could help you to adjust industrially accepted organism to do something else, something that you wanted to do. And being able not only to model, but to really create a modified organism with desired property. That probably concludes the part to illustrate our approach to look for bioparts from which we can compose different types of machinery, including biorefineries. And I would like to say a couple of words about the final chapter, which is focused on diversity. There are estimated over 1.5 million of different species of fungi on this planet. Very little is known about them, very few well characterized, and there is almost no genomic information in comparison to bacteria and other organisms. On this picture, you see some of examples of them growing in nature. Many of them cannot be cultured. And on the other hand, you see measured groups of fungi that cover this 1 million+ different species and how they evolutionarily relate to each other. The goal of this chapter is to understand both ecological and phylogenetic diversity of fungi. If we think about wood decay, I just mentioned how we study individual organisms to see what genes are encoded there that enable this wood decay, how we can use model organisms to validate some of these functions. But if we think about nature, how wood decay happens in nature is probably through succession of different organisms. One doing some function, another organism comes after that, and maybe together with other groups of organisms, fungi or prokaryotes, decaying certain elements of leaf litter and wood material. So alternative approach, instead of looking at function of individual genes, instead of looking at individual genomes, we can look at the entire community. And this direction is called metagenomics. And metagenomics for eukaryotic organisms is a little bit more challenging. So it's running full speed for bacteria, where each gene is an open reading frame. And it's a little bit easier to predict genes in sequence bacterial or archaeal genome. In eukaryotic organism, each gene is a combination of exons and introns that are spliced out, and exon form transcript and then translated. predicting those exon intron structures is relatively challenging in eukaryotes, and signals that determine that boundaries of genes, boundaries of exons varies from organism to organism. The data produced by metagenomics of relatively complex community, we're talking about relatively short reads produced by Illumina, for example. They're 150 base pairs long. They can be assembled by, if the community is complex, the sizes of the fragments that assembled from those reads are relatively small and often not sufficient even to predict a single eukaryotic gene. So therefore, in order to do metagenomics for eukaryotes, you need a lot of reference genomes. The genomes with which you can compare your fragments, your assembled fragments, and determine which of these organisms, or close relative of those reference organisms, is present in your community. And one of the projects that we have, again, collaborating with Francis Martin from Nancy from France, is to look at several forest sites, look at diversity of fungi in each of them, and not only identify composition of fungi that grow in those forests, but maybe try to isolate and grow some representatives. And basically metagenomics is composition of different approaches at this point. First is a profiling, determining what type of species are growing in your community. And for fungi, the marker is used, it's called ITS, internal transcribed sequence. Basically, you design primers, you use it for all the material, DNA materials that you get, and you identify a diversity of different species you find there. Metagenomics, I mentioned, is challenging, and we need a lot of reference genome, and you can produce reference genome for the community that you're investigating by growing isolates. Or we can look at metatranscriptomics, where we overcome the issue of predicting complex gene structure and looking at the transcripts. It's already spliced and equivalent of open reading frame, and look not only at the composition of species, but at functions, at genes that are mostly expressed, at function and dynamics of those functions within a community. But here again, ideally you have a reference set of genomes. And you can produce these references within the context of individual projects, but you can also generate a general framework where, and that's lead to my next topic, the project called 1000 Fungal Genome, project led by Joyce Patafora from Oregon State University, which is a collaboration of 15 different labs and culture collection, with the idea to produce in about 5 years sequences of about 1,000 genomes. And it's not the number that is important here. So the goal is to cover the whole diversity of fungi that exist in nature. And as an organizing principle, we're looking at the level called families. So there are classes, orders, phyla. There are families of fungi. There is about 500 to 600 different families. And for a small fraction of those, we have at least single genomes that could be used as a reference. So the idea to produce reference genomes for one or couple of genomes for each of these 500 to 600 families. And on this, well, you see tree showing different classes, and within every class you see the bar reflecting diversity. So for example, you have about 100 families in Basidiomycetes or Agaricomycetes. There are classes that are not as diverse, but still very little known about them. Green color indicate families with no genomes. Blue indicate groups where we have genomic information, and red are those that are in progress. So you see that majority is green. We need to sequence that. And while we're working with culture collections, we open this project for entire fungal community of fungal biologists. Everybody around the world can go to our site, microcosm site, click button, nominate, and nominate species for sequencing in those green area. For the families where there is no sequence information, you can suggest sequence some species for which you can provide DNA RNA material. So hopefully through these efforts we can build a much more comprehensive reference data set for further metagenomic studies, for better systematics, for phylogenomic studies and other applications. And since I mentioned one of the functions of microcosm site, I would like to also illustrate some tools that are involved in analysis. So through this site, which is once again available to anybody at gig.gov/fungi, which currently have a collection of 250 fungal genomes, you can click on any nodes of this tree, which on one hand illustrate fungal mycelium, hopefully, on another illustrate phylogenetic relationships between different groups of fungi, and in red the groups of fungi for which we have some sequence genomes, in gray those that remain to be explored, or at least they are not public yet or not published genomes. So you can click on each of these nodes and ask questions related to smaller groups, or travel through the tree and ask questions related to all fungi. For example, find genes among all 250 fungi using either keyword search or blast analysis, or download those sequences. So I mentioned function of nomination. You can add to this tree. We get DNA RNA sample, we sequence and annotate them, they will be added to this collection of 250. And then you can explore those genomes in two principal ways, either through genome-centric analysis, focusing on that particular organism, or through comparative analysis. For the nodes where you have more than one genome, you can compare them side by side. The heart of genome-centric analysis is Genome Browser, which displays genome sequence, scaffolds and contigs of assembled trees along horizontal axis, and different features found or predicted in those scaffolds or contigs. For example, the first track on this screen you see is GC content, number of G and C that illustrate composition, nucleotide composition of that organism. So in the red you see gene model track, where you see all the predicted models predicted by different algorithms, sometimes varied from one genome to another. We're trying to find optimal solution for each of them. In green, you see expressed sequence tags, ESTs, transcriptomics data mapped to the very same assembly. And in purple, regions of homology or similarity on a protein level between genes predicted in these genomes and genes from other organisms. This browser is interactive. You can zoom in, zoom out, move to the next scaffold, move to the next part of this particular scaffold. Each of these tracks is connected to either external tools or to the tools that allows you to explore function of individual genes in detail. So here's a gene report where you see identifier of the gene, predicted function, the transcript composed of exons and introns, translated into protein sequence in green, for which we predict domains here in orange, for which we identify also homologs by running BLAST or other types of sequence alignment. And all these labels and images are interactive and allow you to get more information. One of the important features of GGI browser, of GGI genome-centric analysis, that for every genome that we sequence and annotate, we also offer tools for manual curation. Community formed around each of these genomes or groups of the genomes can go to a website from their offices and edit some of the function, add functional information, add data, refine the structure of predicted genes. And principal investigator of each project usually coordinate activity of this group, and well, usually welcome new members of the group. So if you're interested in a particular pathway, you can contact us, contact PI, and join the efforts. The more the merrier. And exploration of these genomes require significant efforts and a lot of manual curation. So here's an example of genes curated by one of our collaborators who assigned function and added some notes. In addition to that, there is a set of comparative tools that allows you to align genomes one against another. Here you see list alignment translated, DNA, whole genome level DNA alignment translated into dot plot, which indicates regions of so-called mesosynteny, where you see, with the dots, you see genes that are shared between two organisms and their order, genes same between chromosomes in different organisms, but not necessarily in syntenic order. At the bottom, you see a sample of the cluster or gene family, where all the members of the cluster can be compared side by side, their gene structure, their domain composition, their local synteny or function of the genes that surround them. Each cluster can be then translated into a tree. Large-scale statistic number of genes in particular families can be compared side by side, and we functionality genes according to Go, Keg, and Koch classifications. To summarize what I mentioned, within a fungal program, we ask various scientific questions. We're interested in interactions and between fungi with each other and plants and their evolution. We're interested in more applied parts, developing a bioparts list for building some machinery for biorefinery. We would like to explore complexity of ecosystem and diversity of fungi. We are user facilities, therefore we welcome proposals from different research groups, and to give organizational principle to different types of proposals, we have this project of Genomic encyclopedia, where we're looking at the groups of fungi, 1,000, and for which you can nominate genomes for sequencing to organize a new chapter of this encyclopedia. We have a project 1000 genome where you can nominate new species for sequencing and send us samples basically all year round. And we started efforts on functional genomics, and we encourage collaborations with different groups. For all the sequenced genomes, we have developed tools for writing proposals, submitting them, sequencing, assembling, annotating genomic data, and exploring them, as well as supporting this exploration using web-based tools. These are different opportunities through which you can interact with GHI, propose something to sequence, develop new technologies, or just simply visit us. And I also would like to mention the meeting that happens next March, one of the largest genomics meetings devoted to energy and environment, in March 18 to 20 here in Walnut Creek, when you can see the facilities and hear interesting stories that come from genomic exploration. So at this point, I would like to conclude my talk and acknowledge GHI staff, all our users, and once again refer to the tools that we have. And if you have any questions, feel free to submit them now. I don't see anything yet, but if you have them, maybe not right away, but later, these slides will be available, and my email is on the front page of this slide deck. Thank you very much for your time. Hopefully, I give you a slightly different perspective on fungi, on problems of energy environment, and how we can help to solve them with genomics tools. Thanks a lot.